Are here in person, and for those online, welcome. It is my great pleasure to introduce today our visiting Locke professor, Dr. Priscilla Hsue, who is the Chief of Cardiology and the Chizuko and Nobuyuki Kawata Chair in Cardiology at UCLA David Geffen School of Medicine. Dr. Hsue went to medical school at UCSF and then stayed to complete all of her postgraduate training at UCSF before joining the faculty in 2002. And I learned last night that she was Chief Fellow, which included the responsibility of organizing all of the grand rounds, as Bhavik did as well. So quite a responsibility. It was not long after she joined the faculty in 2005 that she received an email from a young medical student at Ohio State who had seen a webinar for 2005 session at the 12th Conference on Retroviruses and Opportunistic Infections. The topic was cardiovascular risk in people living with HIV. As you probably guessed, that medical student was me. And I can remember thinking at the time that Dr. Hsue must be the most seasoned international expert on a topic that had recently sparked my interest. And in fact, I discovered yesterday when I was reading through her CV that it was actually the first international or national talk listed on her CV. She was already an expert at such an early point in her career. Priscilla graciously responded to that email and allowed me to spend a short research elective in her lab at SFGH, which then led to a career in this field. And that's why I'm here today. My story illustrates the enormous impact that Dr. Hsue has had on science through mentoring and sponsorship of over 70 students, residents, cardiology fellows, and faculty over the years, including one of our own recent fellows, Dr. Sateh Shakil. Her efforts have led to multiple teaching and mentoring awards, including a K-24 grant from the NIH. And as you will see today, her science is also universally recognized as outstanding. She is currently PI on 6 active R01s and co-I on multiple others. She has served on and chaired multiple NIH study sections and is president-elect of the American Society for Clinical Investigation. Finally, she is recognized as a master clinician who has started unique programs such as the Center of Vascular Excellence at SFGH and a longstanding HIV cardiology clinic that has been a model for similar clinics around the world, including ours here at Harborview at Madison Clinic. So Priscilla, welcome. We are very much looking forward to your talk.
Right. Thank you. Thank you. All right. Well, thank you, Chris. That was too kind of an introduction, and I'm so honored to be here. You know, you have so many folks that I've worked with in the past here. So Bhavik, Chris, Danny, and others. So I'm super honored to be here. Thank you. And then folks that I've met from study section, Francis and Kevin. So amazing. These are my disclosures. All right. So how did this story start? When I was in college, the HIV epidemic came out, and I was like, this is so interesting. I want to become a doctor, and I want to go to UCSF. And that's what I did. And I spent a year in a basic science lab with Werner Green. And I think part of the story is I've had amazing mentorship along the way, fantastic collaborations. So I did that. And then around that time, we started to notice there were younger individuals with HIV coming in with heart attacks. And that really stimulated this whole line of investigation for me. So I'm going to talk a little bit about first of the description of the clinical problem, and then a little bit about the studies we've done on mechanism, clinical trials, and future studies. And I think there are some trainees in the room, people in fellowship, maybe others. I want to say that I've failed many times in my career, and I think the important thing is to love what you're doing, to always have that resilience and persistence, and that will help you on your way for this kind of career. So I know Chris knows all this, but I'm guessing some of the folks do not. This is data from UNAIDS. 91 million individuals have been infected, 41 million living with HIV, and 44 million have died. Of course, in the United States, over 1 million individuals living with HIV, 39 new diagnoses per year. And these disproportionately occur in the South at higher rates in African American and Hispanic individuals. And it's crazy to think that it's been over 40 years of living with HIV. So what are the medications? What are the recommendations? And these came out last year, and this is for our cardiology colleagues. So when to start HIV medication ASAP, what to start. The first line is now integrase inhibitor therapy, and usually 1 tablet once a day. And what are the goals? They would like to have 95% aware of their diagnosis, 95% on therapy, and 95% treated and suppressed. And where are we? So we're closer, but not quite there yet. So there is room to grow. What about cardiovascular disease? So if you look in the literature, the first case described was in 1990. If you look at that, and fast forward to 2018, 28 years later, people with HIV are twice as likely to develop cardiovascular disease. That burden has actually tripled in the past 2 decades. And I think it's not one size is the same everywhere. So in developed countries, it's more athero. And then Chris and others are doing work in underdeveloped areas, and it may be a different kind of cardiovascular disease. So we have lots of take-home points, and I'll try to get to these. So the HIV population is aging. Chronic inflammation, immune activation are strongly predictive of events. I think as cardiologists, HIV is a model for chronic inflammation that can help us identify therapeutic targets in general. And vice versa for our HIV colleagues, targeting inflammation can actually have an impact on cure. We're also using HIV as a model to study new viruses. I'll also touch upon some biologic and gene studies in HIV and end up with future studies. So I want to talk about description of the problem, which is how I started with things. So when I started San Francisco General, there was not much infrastructure to do research, so I got advanced training to do imaging, set up a team of mentors across specialties, building a cohort. And that's probably about the time I met Chris. And this really led to this thing called the Center of Excellence in Vascular Research. It's using non-invasive imaging, and it's in addition to IMT, using FMD, endopat, other imaging, more recently CPET. It supported many different investigators since that time, including Jamie McCabe, I know, who has spent a lot of time at UW. So it takes time to develop clinical events. And so a lot of the first studies showed atherosclerosis and cardiovascular disease in HIV using imaging. And this is a carotid ultrasound, but you can, whether it was CTA, coronary plaque, or CAC, this was found by our group and many others. And in fact, sometime after that, studies have begun to show that individuals with HIV are at high risk for acute myocardial infarction. This is data from the VAC study showing 50% increased risk of acute myocardial infarction, strong risk associated with HIV, similar to that of super potent traditional risk factors like hypertension and diabetes. And you can see here, even among younger individuals. So I will say the first part of the work that my group and others have done was to establish this field, looking at atherosclerosis, heart failure, arrhythmias, and I'll come back to that, pulmonary hypertension, stroke, and many other sequelae. So I think at this time, this slide is to show that I'm getting old enough that my mentees are mentoring other mentees of mine. And this is also to show that I'm very proud that we got 3 interventional cardiologists to study HIV and cardiovascular disease, which is really saying something. But these are more recent studies done by mentees of mine. And then Katie Kentafia, who's now at Beth Israel, was mentored by Eric Szymski, who's one of my initial mentees. And so that has been thrilling to see my grand mentees move on. So the HIV population is aging. If you look in this Dutch cohort and flash forward to 2030, which is just a couple of years from now, it's anticipated that 73% of individuals with HIV will be aged 50 and older. And to that end, and Chris alluded to this, at the beginning of my career, we started specialty clinics looking at cardiovascular disease and HIV. This has evolved with time. There's now something at San Francisco General called the Golden Compass. This includes like HIV neurology, dermatology, geriatrics, and it's really reflective of the aging of the population of HIV in the United States. And you can see actually individuals between the age 55 and 64 make up 26% of the total. And these have served as a nice prototype for clinics across the world. So I want to go now on to mechanisms. And so I think at the beginning of this, everyone was like, it's all traditional risk factors. It's blood pressure, it's smoking. And so clearly those are an important contributor to this. These are data showing, this, there's many other studies. I think it's interesting, this Danish cohort study showing losing more life years to smoking than to HIV infection. And that should be an important reminder to all of us. And what about antiretroviral therapy? So this itself could take up an hour, but I'm going to just breeze through it. Protease inhibitors are associated with cardiovascular risk. It might be related to dyslipidemia. Newer agents like atazanavir may be protective. Abacavir, which is an NRTI, recent use is associated with higher risk of cardiovascular disease. So the guidelines suggest that you should probably consider switching individuals on this agent. And then, of course, now the first line of therapy, which is integrase inhibitors, are associated with weight gain. And so how is that going to affect cardiovascular risk? And so as the types of HIV medications have changed, so have the types of cardiovascular complications. So at the beginning, before there was any kind of HIV medication, we saw effusions, dilated cardiomyopathy. I've talked a little bit about protease inhibitors, and this is when we're seeing reports of atherosclerosis and dilated cardiomyopathy. We now know that in integrase inhibitors, maybe more arrhythmias and heart failure. And of course, as we look towards the future, maybe they'll cure HIV, there'll be newer agents, and what will the side effects be associated with that? So now I want to turn it back a little bit to HIV disease itself. And this was done by Jen Ho, who's the first fellow at UCSF I ever worked with. And she did a whole line of studies showing that aside from HIV medication, traditional risk factors, that having poor control of HIV disease, as well as advanced immunodeficiency, were strongly associated with atherosclerosis. This has been now shown to predict clinical events as well. And this has led to a whole line of investigation showing that even in the setting of treated and suppressed HIV infection, there's chronic inflammation, immune activation. These are in turn strongly associated with clinical events. And yeah, it's interesting. This has led to now many different studies. And aside from cardiovascular disease, it has turned now to a focus on inflammation as a way to cure HIV infection. And I will circle back to that at the end of the talk. So now I want to talk about 3 studies that have broadened our understanding using imaging, cohorts, and biomarkers. So we have used imaging as a way to ascertain pathogenesis as well as the impact of different interventions. And so FDG PET, it's a metabolically active glucose substrate that gets taken up by inflammatory cells and macrophages. This is a study by Amit Tawakal, one of my collaborators, showing an individual with low uptake on the top and high uptake in the bottom. And using this, we studied both arterial and hematopoietic lymph node activity using arterial inflammation to be able to do this and look at the impact of HIV-associated factors. And so doing this, there's an uninfected individual on the top, and on the bottom, you can see that those lymph nodes just really light up in the setting of HIV. And there was a nice graded increase between that activity in those lymph nodes and degree of HIV disease control. And using some specialized persistence assays that can look at less than 1 copy per mil, we found that there was a strong association between assessments of the viral reservoir and imaging of these hematopoietic tissues. And so this is one of the first studies to really quantify the reservoir and associate it with imaging. And I think if you want to play devil's advocate, you can say that's really nonspecific. So the group at UCSF looked at a more HIV-specific marker. This is VRC01. It is a link to the HIV envelope protein, radiolabeled with zirconium, taken this through animal studies, first in human, and now studying them both at Davis and at UCSF. So it has been studied in humans. And we coupled this with the ability to study tissues at UCSF. This is a gut biopsy program that has been developed. They've done almost 1,700 gut biopsies in persons with and without HIV, with and without COVID. They've also done lymph node biopsies and lumbar punctures to get cerebrospinal fluid. And this is really an amazing program. And so coupling these two together with my collaborators, we looked at this HIV-specific tracer. You can see negative individuals and a viremic HIV individual on the right-hand side. And then I think it was great, coupling this with gut biopsies and gut assessments of the reservoir, we were able to show a strong association between those tissues assessments of the reservoir and this imaging. And so we're now looking at this in the context of different interventions, whether it's HIV medication or different anti-inflammatory interventions. So what about sudden death? We have Babak, who's our EP colleague in the room, clearly a terrible complication. And so this came up, Zian Seng is my colleague at UCSF who was studying this in San Francisco. And in the first couple of patients he noticed had HIV infection. So he called me up and he's like, what's the deal with this? Has this been described? And it turns out it had not. And so this is an older study. So from 2001 to 2009, you can see that AIDS deaths were more common, but what was surprising was that sudden cardiac death, shown in red here, was more than 4 times the general population. We really didn't know why this was the case. And so this led to a whole study within the context of Zian's study looking at sudden cardiac death in HIV in San Francisco. This is an amazing infrastructure that he's built. So everyone in San Francisco who meets the criteria of sudden death gets a death certificate review. There's toxicology. We talk to paramedics, and then we have access to tissue. And you can see in this time period, there were 109 individuals with HIV who met this criteria, and 108 got autopsy. I mean, that is like amazing and a ton of work. So here are some of what we found. Long story short, a lot of these individuals died of occult overdose. And of course, like a defibrillator is not going to save those individuals. But we did have occult cardiovascular disease also in those individuals. And this is a messy diagram, but this finding of both sudden cardiac and sudden arrhythmic death that was adjudicated by autopsies was driven by white men. I hate this diagram, it's like too hard. But I think the fascinating part of this was that access to heart tissue. And so here you've done trichrome staining for fibrosis. And again, long story short, the fibrosis was higher in the hearts of everyone living with HIV, or I guess who had been living with HIV. This was driven by interstitial fibrosis and not replacement fibrosis, which is what happens in the setting of cardiovascular disease. And we believe that that fibrosis was driving some of the arrhythmic findings in the study. So this did not make the paper, but I thought this was super interesting. It's nanostring expression profiling in these individuals. And you can see here that some of the markers of macrophage activation, T cell activation were associated with fibrosis in the hearts. So I thought that was super interesting. So I know we have Kevin here from heart failure and others. What about heart failure? So again, using the VAC study, both HFrEF and HFpEF have been higher in HIV. It's more common in poorly controlled HIV infection. Some studies showing that it might be associated with protease inhibitors, but not all. And 2 former fellows with my group who are now heart failure docs at OHSU, Nalini, and then Teresa at University of Pennsylvania, looked at the heart failure network, and they found that markers of gut issues, TMAO, were strongly associated with myocardial fibrosis in the heart failure network. We know that HIV preferentially infects CD4 T cells of the gut, and so this whole microbial translocation and leaky gut could be a way that HIV leads to heart failure. We have also partnered with, I really wanted to get at this at a more molecular level, and I'm not a basic scientist, so we partnered with individuals at UCSD. And the last GIF is an amazing cohort. It's basically individuals living with HIV who pre-donated their bodies to science, and then after dying, within 5 hours, they get a full autopsy. And so we've partnered with Davey and Sarah at UCSD and Neil Chi, who's a basic scientist. And then at UCSF, we've also started banking heart specimens for both LVAD and cardiac transplants. And we've now collected samples on over 60 individuals, several of which have HIV infection. So what have we found so far? And this is some preliminary data using single-cell RNA-seq. First of all, we found that our samples were good enough quality to be able to do this molecular analysis. This is an individual with and without HIV. You can see here there's contributions from lymphocyte, NK cells that may be different. And working with Neil Chi at UCSD, we will be doing kind of disease in a dish model, human pluripotent stem cells, to model the impact of HIV medication and chronic inflammation in this cohort to try to get at this in a more molecular sense. As a clinician, we want to be able to identify who's going to be at risk. And I think the issue, and this has been shown in tons of studies now, that traditional risk factors do not do a good job of estimating risk in HIV. And it may particularly underestimate risk. This has been now shown using a bunch of different cohorts, a bunch of different calculators, and this includes the pooled cohort equation. So with this, we wanted to look at different approaches to be able to risk stratify individuals. And so proteomics, unlike genetics, can change the reflective environment. They can also tell us a little bit about disease processes. And in the general population, one of my mentors, Peter Gantz, has used this to improve risk calculation. So we use this approach to study risk prediction HIV, and this was in the VAX cohort, to be able to identify proteins to improve our risk in HIV infection. It's a large cohort for HIV, around 1,500 individuals and 853 matched controls, and they have clinical events. And these are some of the proteins that we identified that were strongly predictive of mortality. And you can see here, the ones that are positively associated with mortality are shown in white. The ones that are protective are shown in yellow. And a lot of these have known therapeutic targets, as shown on right. So hopefully that's something that could be explored in the future. And these proteins stay strongly predictive, even if you put in things we know that are strongly associated with mortality, like CD4 count, viral load. They remain strongly predictive even in the setting of all these biomarkers. And over time, Kaplan-Meier curves in both the training and testing cohorts showed that they're strongly predictive, better in early years. So this is one of the initial studies to look at proteomics, identification of unique proteins. And I know Chris highlights SATE. So SATE has really picked up on this and used these proteomics to predict incident heart failure in HIV. This is, again, a busy slide, but to highlight in green here, you can see that they're strongly predictive of heart failure. The proteins only, better than adding clinical risk factors, better than adding CD4 viral load, better than addition of inflammatory markers. So she's currently writing this up. And she presented this summer. All right, so I want to go on to the trials that our group has done in different diseases. So I've told you all about high risk HIV cardiovascular disease. So we're in cardiology. What are we going to do to reduce that? What about low-lying fruit, statins? Okay, so before the Reprieve study, what were the recommendations? And that is individuals who had HIV, who had this borderline risk between 5 to 7.5%, could be a risk enhancer. You could use that as data to start that moderate to high intensity statin. And if you're still on the fence, maybe you could do imaging using CAC. Okay, so the Reprieve study, which was a study of pitavastatin and HIV, was published 2 years ago, over 7,000 individuals. And you can see here, age 40 to 75, complicated inclusion criteria, so risk up to 15%. There was a significant reduction in cardiovascular events. And so I have a question for the audience. So, okay, I've shown you those results. Does that mean everyone with HIV should be on a statin? Yes? No? Okay. It's too early. Everyone's tired. Okay. So this is my personal answer. I don't think everyone, but I think it does suggest that we should have increased use, particularly among those who have a calculated risk of 5% and higher. And so why do I say that? So I look at the number need to treat. What is that? So that is the number needed to treat for using a therapeutic agent to reduce one event. So the lower is the better. If we look at the statin guidelines for ACC and the U.S. Preventive Services Task Force, we see that the overall Reprieve result was around 102, and that would be in the maybe not. But in individuals who had a low risk score, you can see the benefit is less. And among individuals who have a higher risk, the number need to treat seems to be more favorable. And so using those guidelines, I think individuals who have that risk, around 5% would probably be indicated. And of course, last year, the guidelines really reflected to change this. The British HIV and European Society says, okay, everyone with HIV over the age of 40, you should think about statins. And of course, in the United States, they're a little bit more conservative. You could think about it among individuals who have that lower risk. Anyways, does having that low risk, if you have HIV, mean you aren't going to benefit from a statin at all? And so I will say imaging studies show that this may not be the case. And so Fan, who had been here, looked at using guidelines for statins, and he found that among individuals with HIV who were not recommended on statin, 2/3 of them had evidence of plaque. And other imaging studies show that up to 30% of persons with HIV who have a low calculated risk may have evidence of plaque on imaging. And so I know as cardiologists, we hate to say this, but I do think among some individuals that even among asymptomatic individuals with HIV, if you're on the fence about a statin, you can think about doing some kind of imaging. So we know in the general population, if you have cardiovascular disease, everyone should be on high-intensity statin. Some will benefit from lowering cholesterol, some will benefit from lowering inflammation. Let's talk about that in context of HIV infection. So I want to first talk about the cholesterol risk. And so before the REPRIEVE study, and this was done by Megan McLaughlin, looking at patients at UCSF, she found that among 43% of individuals that were eligible, 44% were on statin. And so that means 50% of patients who should have been on statin were not on them. And so I do think the REPRIEVE results are an important reminder for all clinicians to think about statins and HIV. Even before the study, they were undertreated. This, of course, has been shown in other studies, and you can see here individuals eligible for statin in this large NA Accord. This is around the 2014 guideline. Now the guidelines have shifted that more individuals should be on statins. How will that statin gap change? So that's something to think about. So we know statins are good, but they're only going to be good if persons with HIV get them, take them, and stay on them. And so we know in the general population, both primary and secondary prevention, around 50% of individuals who have cardiovascular disease are not on statin. And so statin persistence is low over time. And this has been studied in HIV infection. And so will that adherence to statin therapy impact persons with HIV? So I want to now move on to beyond statins. And so we know PCSK9 inhibitors, injectable medications that reduce LDL cholesterol significantly once every couple months, and now potentially once every 6 months. And so I want to put up to the audience, so we know that inclisiran, there's now outcome studies there's outcome studies going on in primary prevention. Can we go along with the success of injectable antiretroviral medication in HIV and couple that with injectable lipid lowering, maybe even injectable antihypertensive medication in the future to improve outcomes? And so Payal Kohli, so I know Babak worked with her at UCSF. She was a fellow in our group. She looked at PCSK9 levels in HIV infection, finding that they were higher. They mirrored levels of inflammation. And I want to think about, in the general population, what do we know about PCSK9? And this is Fourier Olay. And both of these arms got PCSK9 inhibitor. I want to say the only difference is one of them got them sooner, and that showed a significant reduction in cardiovascular mortality. It did make me think about, should we target a lower LDL goal in the setting of HIV infection in this context? And in fact, I think maybe in HIV, we should think a little bit more about the benefit of lowering LDL earlier. And that is, I like this diagram, because you can see, okay, at age 50, if we lower LDL, here's the clinical benefit, and this is in the entire population. But if we start LDL lowering earlier, you can see, before the age of 50, you can see there are additional clinical benefits. And if we do that, will anyone even care about inflammation? And should we start thinking about lifetime risk in HIV instead of a focus on like 5-year risk or 10-year risk? And that, I think, is something to think about. So our group has finally finished a study looking at PCSK9 inhibition in HIV, lowering cholesterol very low, and the impact on arterial inflammation, on non-calcified plaque. And we're also looking at other agents, bempedoic acid. It's an inhibitor of ATP citrate lyase. In the general population, it has reduced events. We are also studying this in the context of HIV, both at UCSF and at UCLA. So I want to turn a little bit to this residual inflammation on this right-hand side. So in cardiology, there's been 40 years of work on this, starting in the 1980s. Now we have clinical outcome studies. We're pretty new about the role of inflammation in HIV. And to this date, there's no endpoint studies using anti-inflammatory agents in HIV infection. Again, in the general population using statin therapy, we know that the best outcomes are generated when you can get both a low LDL cholesterol and low inflammation, as shown in red. And so based on that, riffing upon that earlier diagram I showed, there may be individuals who benefit from both additional cholesterol lowering and lowering of inflammation as well. So you could play devil's advocate and say, well, all those studies were done using older statin medication. What is that residual inflammatory risk? And so here is using high-intensity statins as well as PCSK9 inhibitors. If you look at orange and red, you can see that residual inflammatory risk remains high, even using modern lipid-lowering agents. And so in this diagram, if you look at reduction in LDL cholesterol on the x-axis and relative risk reduction, we have all these lipid-lowering studies. We now have the study in HIV, which falls nicely on this curve. But we have a bunch of studies that didn't touch cholesterol, but they lowered clinical events. And so based on that, the studies to date in this large HIV studies showed that statins did not seem to significantly impact inflammation. And in that clinical outcome study, among individuals on the statin arm, 40% had clinical events, showing that there is still a role potentially for additional risk reduction in HIV. So to take a moment, we've talked a little bit about toxicity for antiretroviral therapy, microbial translocation, immune activation, and then the fact that antiretroviral therapy controls HIV disease, right? But it doesn't cure it. And so because of that, all these factors come together to cause chronic inflammation, which are associated with comorbidities. And in multiple different cohorts using multiple different markers, this has been shown. And it's pretty remarkable that this has been consistently demonstrated across the field, that these markers are strongly linked to clinical events as well as cardiovascular disease. So, with that in mind, if we could lower some of those markers like IL-6 and D-dimer, this could translate to an improvement in clinical events as well as mortality. Although we're talking about cardiovascular diseases today, inflammation is strongly associated with all sorts of head-to-toe comorbidities in HIV, cancer, premature aging, kidney disease, and HIV cure. So will statins get us there? Will statins lower inflammation in HIV? And so I will say the studies to date, which were a bit smaller, have been a bit mixed. And so we don't know if they're going to target the pathways we think are important in HIV infection. And in a sub-study of Reprieve, they found that statins significantly reduce LDL cholesterol and reduce plaque progression. But interestingly, there's a much more modest effect on inflammation. Sorry, I'm the cursor is being fast. Okay. So I want to talk a little bit about different anti-inflammatory strategies that have advanced the clinical testing. And so we're going to talk a little about broad immunosuppression, low-dose methotrexate, versus laser-focused hit on inflammation, canakinumab, targeting innate immunity, and then we'll go back to colchicine. So I'm going to go over a lot of studies here. I'll try to make it clear. So in the field of HIV, we looked at low-dose methotrexate and to look at the safety and the impact on inflammatory markers. So long story short, there was a small reduction in CD4 T cell count, which is not good. There was a significant decrease in CD8 T cell count, which is it is a target for HIV cure, and didn't touch inflammatory markers or coagulation markers at all. Now, if you go to low-dose methotrexate in the general population, this was, of course, studied in CERT. There was no impact on inflammatory markers, no impact on events. Okay, so we've talked a little bit about statins and low-dose methotrexate. What if we go higher up in the inflammatory cascade? What if we target IL-1 beta, which is impacted by cholesterol, neutrophil extracellular traps, turbulent flow, and canakinumab is a monoclonal antibody to target IL-1 beta. So we did a small proof of concept study in the setting of HIV infection to look at safety and impact on inflammatory markers and imaging. We found that using traditional markers, it did not seem to impact CD4 count or viral load. There was no impact at all on T cell activation. However, there was a significant reduction in inflammatory markers, as well as a significant reduction in both arterial and hematopoietic activity. So now if we go back to the general population in CANTOS, of course, this was looked at. There was canakinumab reduced inflammatory markers. It did not touch cholesterol. It reduced cardiovascular events. But due to complicated reasons, it did not move on for regulatory approval. So what about colchicine? I think this is a surprise to all of us cardiologists. Not sexy, but at least in some studies, it seems to work. We know that after MI, colchicine reduces events. In the setting of chronic cardiovascular disease, it also reduces events. It's the first anti-inflammatory agent that's FDA approved. However, some studies, particularly in the setting of STEMI, show that maybe colchicine doesn't have impact. It has been studied in a small study in HIV that didn't really show much. Okay, so as a cardiologist, these studies that we've done in HIV can inform the larger population. And that is, we know from Peter Libby and many others that both innate immunity and adaptive immunity come together to cause atherosclerosis. From our studies in HIV infection, we think that IL-1 beta inhibition may impact innate immunity with the impact on inflammatory markers, while low-dose methotrexate may have an impact on CD8 T cells. And I think these are studies that we learned from the HIV population, and so that's important. There's also many overlapping pathways between atherosclerosis in general. So you have monocytes, macrophages interacting with the endothelium, causing occlusive thrombosis. Also, these similar pathways in the setting of HIV infection. And of course, this can inform us about how immunology interplays with cardiology. And the population with HIV, they're incredibly engaged, interested in studies. And so this is a great model to study. So we talked about inflammation and cure at the very beginning of this talk. And I want to say we don't know, it's a chicken or egg issue, right? We don't know if inflammation causes the virus to persist or persistence causes inflammation. So if you cure HIV, that's one way to do it. The other way, you could try to lower inflammation using agents and see what that does to HIV cure. And so using sirolimus, this is an mTOR inhibitor. Our group studied this in the setting of HIV infection. There were significant decreases in CD4 T cell associated HIV DNA. And interestingly, this persisted even after stopping the drug. And Megan McLaughlin, who was a fellow with our group and is now a faculty at UCSF, looked at the association between viral persistence and carotid plaque, showing that there's a strong association between these indices and atherosclerosis. So therapies that might impact the reservoir might help us reduce cardiovascular risk. So I also want to be humble and to say that, we think that HIV disease is at the bottom. There's inflammatory, inflammasomes, macrophages in the middle, inflammation and comorbidities as the branches of this tree. I think we have to be humble and see that if we intervene in one pathway, it can have unknown sequelae, and it's probably a lot more complicated. And so I think canakinumab was very humbling to me as a lowly cardiologist, and immunology is very complicated, I think. And that is, I talked about canakinumab and cardiovascular disease, but there was a very strong association between canakinumab and fatal cancer, cancer incidence, and lung cancer. And I really didn't understand why that would be a case. And so is there some way that if you reduce inflammation, can you have an impact on counter-regulatory immunosuppression, which could lead to better control of cancer, better control of HIV disease? And this is from Peter Hunt at UCSF, and that is inflammation, I think, of the gas. The body is stepping on the gas, and it's bad. But what your body doesn't respond to, it tries to step on the brakes, and that's immune regulation. That can impact checkpoint inhibitors or receptors, suppressor cells. And those immunosuppressive responses, stepping on the brakes, could blunt responses to both HIV and cancer. And that is that canakinumab, using our trial, Rafiq Sekali and Jeff Tomalka, have shown that a certain subset reduce their measurements of the viral reservoir getting canakinumab. And using bulk RNA sequencing, they found that IL-1 beta inhibition improves T cell function, which was associated with lower indices of the reservoir. And so that was good for the heart and may be helpful in the setting of HIV. But I also want to play devil's advocate. That is, what is good in HIV or cancer could actually be bad for the heart. And so we know that IL-10 could be protective, along with TGF-beta. JAK-STAT inhibitors reduce inflammation, but they also increase thrombosis. And of course, the very pathognomonic example of this is checkpoint inhibitors. Great for cancer, but there's cardiovascular complications. So I want to now pivot a little bit to COVID-19. So during the pandemic, our group applied all the expertise we had in HIV and immunotherapies, and we used that to study SARS-CoV-2. And so a lot of our initial studies were in the setting of acute COVID infection in the hospital, and this is us consenting an individual on the right-hand side through the door. There's a large team of investigators who did this at UCSF and Montreal. We looked at colchicine in the setting of acute outpatient COVID, canakinumab in the setting of inpatient COVID, and a study of convalescent plasma as well. So colchicine as an outpatient, long story short, didn't work. This was before widespread testing was available. So if you restrict it to those who got a positive test, it does seem to have an effect. It was a very hard study to do. I think what was cool about this, it was totally contactless. So patients signed consent using through the internet, and then the colchicine or placebo got couriered to them. So I thought it was really cool. Canakinumab in the hospital didn't seem to have an effect. Again, if a lot of these individuals got other immune modulators like tocilizumab, so if you looked at people that only got canakinumab, maybe somewhat of a difference. And then during this time, there was this thing called long COVID that came up. We started hearing about it in the summer, maybe. And then there was some more attention brought to this with NIH funding as well. So this is a continuation of symptoms after more than 3 months after the acute infection. And so it's very common and is similar to other long syndromes. And what's the conceptual framework to this? There's lots of, I think, parallels to HIV. There's inflammation, viral persistence, causing downstream pathology. And so I want to highlight this for the junior investigator. So Michael Peluso, who's a junior investigator at UCSF, he leapt to this during the pandemic. He established this cohort of, we didn't even have a name for it at that time. So it was long-term impact of infection with novel coronavirus. And so now there's over 1,700 individuals, banking of biospecimen. There's a tissue program and an imaging program, and now clinical trials. And it's amazing if you look at the samples that he built from this cohort are now used internationally. And it's a great, I think, model for all of our junior investigators. So just like in HIV, inflammation is consistently identified, as well as SARS-CoV-2 persistence. So during the pandemic, we looked at our ability to get gut tissue, and this is some cool work. So we did gut biopsies on individuals before the pandemic, as well as long after acute COVID infection. And this is immunohistochemistry showing actively replicating SARS-CoV-2 among individuals, even now, like up to a year after COVID, or more than a year. And so that should only be made during active replication. So we're trying to see now if that's associated with symptoms, and so stay tuned. Matt Dersenfeld, who's a junior mentee of mine at UCSF, decided to look at cardiovascular sequelae of long COVID. And so he set up a CPEP program and did lots of imaging. And so this is one of his first studies looking at this. So he did MRI, didn't really show any differences. He did Zyopatch, we didn't find anything. So he did find that pericardial effusions seem to be more common in the setting of SARS-CoV-2 that were associated with inflammation. And what he did find that when setting up a CPET program that this was where this seemed to differentiate and underlie a lot of the findings that we saw among individuals with persistent findings. And this is a meta-analysis, but he also did this in the setting of long COVID, showing that VO2 consumption was reduced. This seemed to be driven by chronotropic incompetence. He's also now demonstrated this in the setting of HIV infection. We now want to think about, okay, that's great, but how do we treat long COVID? You know, is it going to be antiviral therapy, immunotherapy, other agents? And so kind of building upon what others have done in acute COVID. So baricitinib is a JAK kinase inhibitor. It is an immunomodulatory that's approved for COVID-19. And working with my colleagues, Vince Marconi and Wes Eli, we are doing a study of baricitinib in long COVID. It's 550 individuals looking at cardiopulmonary sequelae as well as neurologic sequelae. And this was first supposed to be at 4 studies, but then the Recover Network has picked this up, and it's going to expand to now an additional 11 studies. So there's going to be over 500 individuals. It'll be one of the largest treatment trials of long COVID to date. So really exciting. All right. So I want to move on to some future studies, and don't worry, I'll be fast. Okay. So we talked a little bit about HIV cure at the beginning of the study. Why is that needed? So 38 million living with HIV, really just a handful have been cured. You can either do that by eradicating the reservoir, or what's going to be easier is probably putting it in remission and controlling it a little bit with HIV medication. So I think a couple of years ago at UCSF, I went to my mentor, Steve Deeks, and I was like, I really want to get involved in some of these gene and cell-based therapies. So at that time, he'd been working with a company to look at T-cell therapy in the setting of HIV cure, modifying these T-cells, and now there was a duo T-cell approach. We tried to start this study at UCSF, and after, long story short, it was incredibly hard, and we couldn't do it at UCSF because the requirement was for 24-hour in-house monitoring. You can't work or drive for 4 weeks. So the trial was shifted to UC Davis. There's been 4 individuals treated so far. There's also been huge advances with respect to gene therapy and HIV cure, and this has been in animals, non-human primates, cleared for first in human. Very intensive protocol for this. So follow it for 15 years. Just think about that. So this was a miss for our group. So we spent like a year on this. We screened 36 patients. It was incredibly hard. So the company ended up going with a local San Francisco person who just does clinical trials, but they have reported first in human, showing no toxicities. So I think I've learned as a non-oncologist trying to dip my toes into the space, you know, a lot of these protocols are set in the context of fatal disease, right? Monitoring for decades, unknown side effects. How do we adapt this in the context of non-fatal disease like cardiovascular disease? And this is coming down the pipeline, right, for PCSK9, amyloid, and of course, lipoprotein a and other studies. And so this is something I think we as a field have to think about. So my colleagues, I think, and I have built this infrastructure to perform this first-in-human biologic and gene studies at UCSF, collaborations with many individuals. We are now trying to start this at UCLA at this California Institute of Immunology and Immunotherapy. So UCLA basically bought a shopping mall and is now like translating it or trying to change it into labs. So in the last several minutes, just want to highlight some work with mentees and others. So cardiology, we hear a lot about CHIP. I know Alex worked with Pradeep Natarajan. So it is a myeloid mutation in hematopoietic stem cells, risk factor for cardiovascular disease and cancer. It's much more prevalent in HIV infection, associated with inflammation and markers of the reservoir. So Alex Bick has reported that it's higher in HRV. Our group working with all these individuals is looking at the prevalence in HIV, the association with clinical events. Is it related to markers of the reservoir and inflammation? And can we look at specific immune cell types that are associated with this? We have also had newer collaborations with global studies. Diane Havler is doing a search study in Uganda and Kenya, really designed to test and treat for HIV infection. It's huge. And they're now looking at comorbidities like hypertension and lipids. We hope this will help to foster global cardiology in the UC system. And I know Chris has done a ton of work in this space as well. Mentioned a little about Sateh Shakil. She was very fortunate to be mentored by Chris. She is looking at unique risk factors for CVD that may include environmental exposure. She's established this cohort in Kenya. And then our group is also now doing some work in Uganda to look at the sequelae of cardiopulmonary disease after TB infection. So we are going to set up a CPET program there with the Uganda Heart Institute. So really excited about that. I just brought this up because of dinner last night. I know we had our great collaborators at Harborview who are doing meth and heart failure. So with several individuals in our group and Jonathan Davis, we tried to set up a cohort to study this, to do cardiac MRI and follow them over time. I'm going to just say this is incredibly hard to do. Patients were died and were sick, and so we tried. So not easy. And then also, I know because Danny and Babak are studying this, Shannon Walker, who's now at Grady, set up a whole genetic testing in the context of meth, cardiomyopathy, and PAH at San Francisco General. Colette DeJong, who's at Stanford, was super interested in this polypill thing. And so what is that? You can see individuals with, GDMT. Can we put that, over-encapsulate that into a tablet and increase adherence? And you can see here, she worked with the pharmacy basically to treat this as an individualized bubble pack. We could over-encapsulate the medication. And she has now finished a clinical trial on this, and very, very interesting work. And so in summary, told you a ton about cardiovascular disease and HIV, told you a little bit about inflammation in the context of two different interventions. We hope that these questions that we learn in HIV and also in COVID can inform cardiovascular disease in the general population. We talked a little bit about cure, cell and gene-based therapies, tons of unknowns. How do we lower inflammation HIV? What are safety issues? How do we move the field forward? This is a statement that's a little bit old, but helpful. Should we have training that's dedicated for HIV and cardiovascular disease? So I want to talk a little bit about my life this past year. So I had been, this keeps going ahead. I had been the chief of cardiology at San Francisco General, which is our safety net hospital. I oversaw 12 people. Okay, so I became chief of cardiology at UCLA. I have 146 faculty. I was looking outside my office on January the 7th. I saw this large cloud. It turns out it was a Palisades fire. There are 28 individuals in Department of Medicine at UCLA who lost their houses. As a new person to LA, spent 37 days with my husband, 3 kids, and dog, not in that order, in a hotel. Was emerging from that, and then we had the federal funding freeze, so $300 million. That was August the 1st. How did that impact cardiology? 28 R01s frozen in cardiology. So that's pretty devastating. So, was there gonna be demands for policy changes? And these grants were restored at the end of last month, and I don't know what the future is gonna bring. But just to close, I just really want to thank all my collaborators and all my mentors, and thanks again to Chris for having me. Any questions, feel free to email me. Thank you.
Wow, that was a lot. And I think something for everyone, really, because you touched
tried.
on the spectrum of translational science from basic immunology all the way to implementation science and clinical trials. So thank you so much.
Thank you.
While people gather their questions in the audience and online, I can start perhaps. So what about social determinants of health? We know that social determinants of health, adverse life events, chronic stress, that can contribute to inflammation.
Absolutely.
So what's your take on that in HIV?
No, I think it's super important, and I think probably not well studied at this point. So I think those are important things, not just in HIV, but I think in the context of drug use, homelessness, and I think all these things likely come together. So I think very important, but poorly studied. Yeah.
Yeah. All right. So I think Babak is up.
Great talk,
Thank you.
as always. I particularly am interested in the study you did with Zian
Yep.
showing the interstitial fibrosis. And I'm thinking about how it got there. It's not replacement fibrosis, so it's probably not macrovascular athero leading to MIs,
Right.
as you pointed out. So in the PET scans you did with Dr. Tawakal's group, those were vascular protocol. I don't know if they had dietary preps for myocardial FTG, but is there any data either from those studies or otherwise on myocardial FTG avidity?
Yeah. Great question. So with Zian, I can't prove it, but if I had to guess, I think it's from, again, we talked about the HIV, it's control HIV, but they don't cure it. So there's still reservoirs in the body. There's ongoing replication causing inflammation and chronic immune activation, which I think ultimately leads to fibrosis, probably not just in the heart, probably in the kidneys, lungs, liver as well. So that would be my guess of how HIV causes that fibrosis in organs. So the studies with Ammittalkal, they were not looking at coronary artery inflammation per se. So we just don't have the resolution for that. So they're looking at arterial inflammation in the carotids and in the aorta. I have not. What was the last part of your question?
Yeah, I guess on those either those same PET scans, or if those patients didn't do the zero carb diet that we do for sarcoid protocol PET studies.
Yeah.
Has somebody or should somebody look at not coronary artery, but straight myocardial FTG avidity, the way we do in sarcoid studies?
Yeah,
Could you look back at those studies and look at the myocardium?
You could. I doubt there was fasting or carbohydrate dietary intake, but it's a great question. Right. I think it would be interesting to look at for sure. Yeah.
I wonder if that would help you identify how the interstitial fibrosis gets there and see a.
It's a great question. Yeah, we don't know.
Agreed.
Fantastic talk. Just to follow up on Babak's question, did that study or any of your other studies had cardiac MRI? I mean, the cardiac fibrosis has been shown in multiple cardiomyopathies to have adverse outcomes. So would that be picked up on cardiac MRI, and was there a difference? Maybe you showed that I missed it.
I did not. I think we only had like an hour time, but there have been studies to look at this. I think first in South Africa, so looking at HIV, done by in South Africa. And then, of course, Chris and others are doing CMR studies looking at different populations to really be able to tease this out. Because I think in that first chart, I said that, cardiovascular disease has tripled. It is in the United States and other countries, it's probably athero. But I think in Africa, Chris and others are showing that it's actually not occlusive atherosclerosis. It could be, something else like myocardial fibrosis, so different types.
Hi, thanks. That was a great talk. So I'm going to ask a question, and I apologize if it's my naivete of the area. I'm not an expert in the area, but it has to do with the whole role of inflammation in cardiovascular disease.
Yes.
It seems inflammation has been associated with just about every single cardiovascular disease. To be honest with you, my first job, I worked with Doug Mann, and we treated inflammation in heart failure and didn't really have that impressive results, actually. Pretty bad results. But I'm just saying despite this pretty universal association, the clinical trials have not really we get a positive trial or a sort of positive, then a negative. And so it really hasn't been overwhelming. And certainly I'm not sure has made it into sort of routine clinical practice. So my question is, do you think the problem is the tools we have to do it? Do you think that the inflammation is really a sort of just a marker of what the actual driver is? And we're suppressing and so because it seems to me the whole field, and not just your area, but is looking at this and saying, we have something here, it's elevated, it can cause disease, but why aren't our clinical trials working?
Fabulous question, and I cannot answer it simply and neatly for you. Not my
All
fault.
Fine.
But no, I think it's a great point, and I think it's heterogeneous, the reasons why. So I think if you look at Cantos, didn't touch cholesterol at all, right? Did reduce inflammatory markers, strong reduction in cardiovascular events. I will say if you look at Cantos and heart failure, there's a suggestion that lowering inflammation might have an impact on heart failure. And I knew that there's some groups looking at that. Antonio Abate's group are looking at different anti-inflammatory agents. So I think it's multifactorial. Like, are we not choosing the right patients? Like, do we need to pre-select based on markers? And if so, what marker would that be? And then I think it's a little bit on what agents are available. So canakinumab, if you think about it, there's no way that can be used widestream like statins, right? It's super expensive. There needs to be lots of monitoring. There's side effects like with infections. And so maybe it's a newer agent that needs to be studied. Of course, Paul Ridker is looking at ziltivekimab, that's targeting IL-6, can be used in the context of kidney disease, and that is building upon, again, the findings of Cantos, in which individuals who reduced their IL-6 by 50% after one dose derived all the benefit, with regards to mortality, MI. And so maybe we just need to be more selective. And of course, newer agents being tested, oral NLRP3 inflammasomes. So I think it's, how do we select who's going to benefit? What's the agent? And, the side effects. It's probably not going to be one size fits all, right? So.
Well, thank you so much,
Thank you.
Priscilla, and we look forward to interacting with you a little bit more today here and at Harborview. Thanks to everyone online. Sorry we didn't get to the online questions. Have a great day, everyone.
Thank you.